Hello, welcome to my lecture series. My name is Gerald de Haan. I'm a molecular stem cell biologist and I lead a lab in Iriba, in which we study how blood cell formation takes place during the lifespan of an organism. In the following 4 lectures, you will learn how blood cell formation occurs, how stem cells play a crucial role in the constant production of new blood cells, and how, and particularly why, blood-forming stem cells can be impaired during aging. In the following lectures, I will discuss with you how the production of blood cells, which we refer to as hematopoiesis, is affected by the aging process. As you know, there are tissues in our bodies in which the rate of cell turnover, or frequency with which cells divide, is very slow. These include, for example, skeletal muscle cells, or neurons in our brain. Muscle cells or neurons that become damaged during the process of aging are not very easily replaced. In contrast, there are other tissues in which cell proliferation is very high. These include, for example, our skin, the inner surface of the intestine, and indeed, also the production of blood cells, which takes place in the bone marrow. Turnover rates in somatic tissue is dictated by the lifespan of individual cells in those tissues. Skeletal muscle cells and neurons are very long-lived, and therefore they do not need to be replenished very frequently. However, cells in the skin and the gut are replaced every few days, and the same holds true for many types of blood cells. Therefore, the aging process is very distinct in tissues with either a high or a low cell turnover. As I mentioned, the blood cells have a relatively short lifespan. This ranges from several months for red blood cells, 1 week for platelets, and only several days for most types of white blood cells. Aging blood cells lose cell integrity and become less functional, and as a result they are removed from the circulation and constantly replenished. Replenishment originates from a very small population of hematopoietic stem cells that lodges in the bone marrow. These are rare cells, and they can, at the single cell level, produce all blood cell types, while simultaneously renew themselves to produce more stem cells. Hematopoietic stem cells are generally believed to be dormant, quiescent, and they live in a hypoxic niche in the bone marrow, where they are in close contact with cells in their immediate environment that provide them with essential regulatory stimuli. Whereas quite some information is available on aging of hematopoietic stem cells themselves, which I will discuss during the remainder of this session, very little is currently known on how the microenvironment in which these stem cells live is affected by age. Hematopoietic stem cells are arguably the best known of all adult somatic stem cells. For sure, they were the first stem cells to be used routinely in the clinic. These cells can be transplanted to patients in which the hematopoietic tissue is destroyed, typically as the unavoidable side effect of high-dose chemotherapy to treat leukemia. Hematopoietic stem cells, as any other kind of stem cell, are characterized by their potential to self-renew. Now, the process of cell renewal dictates that upon stem cell division, one of the 2 newly formed daughter cells displays characteristics that are identical to the mother stem cell from which it originates. Of course, this key stem cell characteristic cannot easily be reconciled with the notion of stem cell aging. If hematopoietic stem cells were to truly self-renew in the strictest sense of the word, then they cannot possibly age. And thus, self-renewal would imply that all molecular components that are present in a stem cell and contribute to its functioning are inherited in a pristine manner in at least one of the 2 daughter cells that emerge after a stem cell division. If one would observe that after replication hematopoietic stem cells lose some of their functional activity, this strongly suggests that true self-renewal is limited, or in fact does not exist. Of note, many clinical hematological syndromes occur preferentially in older people. This relates to a loss of blood cell number or function, such as anemias and immunological problems. In addition, the production of an excess of blood cells in the bone marrow, leading to myeloproliferation or full-blown leukemia, occurs much more frequently in old people compared to young. It is therefore tempting to speculate that a wide range of hematological abnormalities that preferentially occurs in older people, in fact, results from a reduced ability of primitive hematopoietic stem cells to regulate their self-renewal potential properly. For example, if a stem cell would produce a daughter cell with a slightly higher self-renewal potential, that daughter cell on its turn may be likely to generate daughters with an even higher self-renewal probability, such that with time cell clones emerge that have a skewed preference for self-renewal at the expense of differentiation. Ultimately, leukemic cells may emerge that are essentially defined by a high self-renewal and very low differentiation potential. Conversely, if a stem cell generates a daughter cell with a lower than optimal self-renewal potential, such a cell is likely to produce cells with an even lower self-renewal properties, which eventually would lead to the collapse of the hematopoietic system. You have now learned how blood cells are produced by hematopoietic stem cells that possess the unique ability for self-renewal. So the key questions that emerge are whether or not there is a limit to the extent that hematopoietic stem cells can self-renew, and whether indeed aging of the hematopoietic system is caused by loss of control of self-renewal of the primitive stem cells. This I will discuss in the next lecture.